Hi. In the last lecture, I spoke about the structure of the protein, tertiary and quaternary structure. Today I want to continue with the classification of the protein. The first group of the proteins are globular protein. They are round shape, and the other group is the fibrillar protein, fibrillar, which is like a rope shape. They are mainly structural, but globular mainly they are carrier protein or enzymes. And we can classify it for different groups of the globular protein. Then the first type of the globular protein, which has it, they contain the hem. It's very important. As you know, the hem, it is the organic compound which has 4 pyrrole group. Which bind each other. They are a resonance structure. Mainly they have a double bond. Like this. Okay. And in the center there is iron 2 plus with the coordination number of the 6 binded inside it. It has a 4 bond, and the 5th bond it can bind to the histidine, and the 6th bond can bind to the oxygen. Okay? Hemoglobular proteins, they have a hem inside it. Okay, within protein we can have a, for example, I draw it before. It is a myoglobin and hemoglobin. Okay, they are 2 different structures. In the myoglobin, the hem in this structure is in the middle. It has iron 2 plus. It has a 2 helix structure, which called the first one is the F helix. Okay, it has 1 histidine in the middle, which with this histidine can bind to the hem groups, and also they have an E helix. It has also 1 histidine. Okay, they are not in the one plane. The first one, this histidine, is the proximal situation. Okay, and the second one will be distal. This is a distal, and this histidine is the proximal histidine. They can bind from the oxygen from the distal part, and the hem is in the middle, and it is a proximal. The shape will be like it in the 2 axes. Okay, because in the 2 axes we can form proximal and distal way. Okay. And the middle is the iron. Okay, from then we can say from the hem, from the one side can bind to the histidine, and from the other side can bind to the oxygen for the carry oxygen. Myoglobin is mainly we can find in the muscles. As you know, hemoglobin also the structure, it has a hem, but with a different situation, different shape. Mainly carrier of the oxygen in the blood. Okay, they can then hemoglobin we can classify in the 2 groups. Is the myoglobin, and the second group is the hemoglobin. Okay, myoglobin mainly in the muscle. Okay, and they can store the oxygen when they have a less activity, okay? Because as I start from here, for example, in the lung, oxygen go into the blood, okay? Then go to the hemoglobin, okay? Because the pressure of oxygen in the lung is high, okay? Pressure carbon dioxide is the less, because of this Bohr effect. Hemoglobin bind to the oxygen, go to the tissue, for example, the muscle. Okay, lost oxygen, hemoglobin lost oxygen, it become hemoglobin and oxygen. Oxygen go bind to the myoglobin. Okay, and the hemoglobin can bind to the carbon dioxide with water. Okay, which become bicarbonate, and hemoglobin with a proton. Okay, which can, it's here in the lung, in the tissue, or for example myoglobin, oxygen pressure is low and the carbon dioxide pressure is the high. It's very important. If you see here, the oxygen carrying mainly happen by the hemoglobin in the blood. Okay, when the pressure of oxygen is very high near the lung, the hemoglobin can be saturated. Hemoglobin, the structure is not like this. It has a four structure, tertiary structure, which has a 2 alpha, then hemoglobin. It has a 2 alpha, alpha 1 and alpha 2, and 2 beta, beta 1 and beta 2 in the structure. And they become the four of them become the quaternary structure. All of them has the one hem. Then they have a 4 hem. Okay? They have 4 iron. Because as you see here, we have 1 iron, but here we have 4 iron. For example, in the hemoglobin, we have 4 groups bind each other. Alpha 1, alpha 2, beta 1, and beta 2. As you see here, this structure, the number of the hem is the 4, and when they saturated, 1 mole of the hemoglobin can carry 4 mole of oxygen in the saturation position. Okay? When the oxygen binds to the hemoglobin, for example, we have 4 mole. In this case, the structure of the configuration of the hemoglobin change. As you see here, we have a hem. Iron can bind to the iron, can bind to the oxygen. Okay? When we have oxygen, for example, in this structure, when we have, for example, alpha beta like this, okay, it can bind plus 4 oxygen. Okay? And it becomes 2 because it is the first dimer and it is the second dimer because it has a 1 alpha and 1 beta. It is alpha, beta, alpha, beta. Okay? They combine each other and the structure configuration change. Okay? Here we don't have oxygen, deoxyhemoglobin. And the deoxyhemoglobin we usually call T or tau. Okay? The structure of the hemoglobin is the different configuration. Okay? Mainly weak ionic and hydrogen bonds between the dimers happen. Okay? In the weak because and the interaction is very weak between the dimers. And strong contraction and strong force between the inside the dimer. Here is the more strong, okay, but here is the weak contraction, okay. When oxygen binds for these dimers, okay, in this case situation, the bond between the dimers will be very weaker, okay, in the hemoglobin. In this case, the dimers can be a little separate from each other and become this one R group or relaxed. If you see, I can draw like this, a little, for example, weaker here, but here I draw 2, here I draw 1, and R, it is a T tau, and it is relaxed. And in the relaxed form, all of them has oxygen, oxyhemoglobin. And they can easily carry the oxygen. Okay, as you see here in the relaxed or R form, the connection between the dimers is very weak, and because of this, because some of the hydrogen or ionic bond can be broken. This bond is called hydrogen bond and ionic bond. Some of them I go to, for example, 2 here, I put 1, for example. Okay, so it will be weaker. And then the molecule can be a little expanded, a little the configuration change. And it's very important for this case. And as I said, know, when the oxygen pressure is high, in this case, the saturation of hemoglobin is higher. For example, in the near the lung, all of the hemoglobin can be saturated with oxygen. But there is another system can be effect for the binding of a hemoglobin. Maybe you ask me, how is it possible near the lung pressure of oxygen is low? How the hemoglobin lost oxygen? It's very easy, because if the near the lung, pressure of the carbon dioxide is higher, then the pH is the acidic, lower. And according to Bohr rule, we say both say that if in the normal situation, if we like this, if the pressure of oxygen is higher than 100% pressure of oxygen, okay, if you increase the oxygen, it is the saturation if you see here. As you see here it is the lung. Pressure of the oxygen high and saturation. But both say if we have increased the pH, okay, we need more pressure to have the same saturation. Then decreasing the pH decrease the affinity of the hemoglobin to oxygen. Then in the Bohr effect, if we decrease the pH in the Bohr effect, if we decrease the pressure, it decrease the pH. In this case, the affinity of the hemoglobin to oxygen will be decreased, because in the lower pH, the structure of the hemoglobin, as you see, this bond will be configuration of between the two dimers will be more stable, and in this case, oxygen cannot bind easily to the heme ions. Okay, and it is a normal. For example, I can draw this structure. For example, it is a pH around 7.4. Here, pH is 7.2 or 1. If you see, the more shifted to the right. One of the important effect for the hemoglobin. The other things in the red blood cell inside it has a hemoglobin. They don't have a mitochondria. Excuse me, they don't have a mitochondria. And then they don't have a Krebs cycle. Then they can just use with oxygen, okay, with the glucose until the glycolysis. Sorry, then they can use just with the glucose and the glycolysis and got the energy, okay. Then convert to the lactate and can leave the red blood cell. In this case, there is we will study in the later 2,3-BPG, or okay. This molecule is the one of the side product of the glycolysis inside the blood cell, can affect in the regulation of the binding of oxygen to hemoglobin. Okay, it is because it is very important amount of the concentration of this compound 2-BCG or 2,3-BPG. It affects the affinity of the hemoglobin to the oxygen. Okay, when hemoglobin with oxygen bind to the BP is 2,3-BPG. In this case, oxygen change with the 2,3-BPG, and then oxygen go out, and the 2,3-BPG bind to the hemoglobin. Then we can get 2,3-BPG or we can say 2,3-biphosphoryl which is one of the side product of the glycolysis, helps to decrease affinity of the hemoglobin to oxygen. It's very important. But then what we can manage here, for the binding here, oxygen, we can have a Bohr effect, okay? Also the other compound is the 2,3-BPG, 2,3-bisphosphoglycerate, which is a side product of the glycolysis, because hemoglobin, red blood cell hasn't mitochondria, they cannot Krebs cycle, they just stop they got energy with the glycolysis, and in this case, the side product 2,3-BPG can increase, and in this case, the affinity of the oxygen to hemoglobin decrease. It is also affect the second. The first one was with the Bohr effect, and it's very easy. Here is no Bohr effect. Here near the tissue we have a decrease because of the increase the pressure of the CO2. We have a Bohr effect. Also with the second part is the 2,3-BPG, also can affect for the affinity of the hemoglobin to the oxygen. Okay, and both of them can decrease the affinity of hemoglobin to oxygen. And I will explain in 3 lectures later about in the glycolysis about 2,3-BPG, how they can produce and what is the mechanism can increase the production of 2,3-BPG in the glycolysis. And later we can understand very easily which process and mechanism is very important. The last part of this lecture, I will speak about a little about hemoglobin family. As you know, from the hemoglobin family, we have alpha beta subunit of the hemoglobin, but in some situation, in some disease, these subunits can be different, and the patient has a low affinity to carry the oxygen. The first important part is the embryo hemoglobin. In the embryo, when the baby embryo is inside the uterus, the structure of the hemoglobin is different. Hemoglobin has usually between the 110 and 120 days life, but for the fetus hemoglobin, the life of the hemoglobin is very low because the structure of this alpha beta is very different. Okay, and because in this case the life of the hemoglobin is very low in the fetus, and when after delivery, the conversion of the hemoglobin from the fetus to the normal hemoglobin with the degradation, with high rate of the degradation of the fetus hemoglobin, they produce high amount of the bilirubin, and it can cause the yellow skin in the baby, and maybe it is a normal, but in very high amount they use especially lamps and put the baby under it, and this can help to conversion of the unconjugated bilirubin to conjugated, and can easily eliminate by the urine. And the structure of this hemoglobin is different because the genome of the hemoglobin is different. Okay, and because of this, I will continue in the next video this part of lecture about the type of the genome, and also the disease related to the genome and hemoglobin structure. Thank you, and see you in the next part of this lecture.